My name is Morio from Kyushu Institute from Technology in Japan. In this study, I tried to predict outcome, which is called clinical disease activity, which indicates how much the symptoms of a patient is good or bad in rheumatoid arthritis treatment. Rheumatoid arthritis is one of the chronic diseases, and it tends to occur mainly in the elderly people. RA is known that female people have a higher incidence than male people. And since the early 2000s, the use of drugs called biologic products has made it possible to treat for remission. Remission means the cessation of disease activity. In Japan, these 8 biologic products are mainly used: ABT, ADA, CCP, ETN, blah, blah, blah. And each biologic products have different characteristics. For example, biologics which are colored green, this green, commonly act on same factor TNF. However, taken method and side effects are different. So physicians need to consider every biologic product's effect and patient condition. That is a very difficult task. And then we thought that if we could predict patient outcome as a result of biologic product treatment, physicians are able to complete biologic selection easily. So the purpose of this study is predict disease activity in patients with using rheumatoid arthritis dataset. Recent years, there is many studies to predict the response to biologic treatment. Especially Guang and others predicted the response to anti-TNF therapy after basic formulation failure. In this study, Guang used machine learning to predict patient outcome to support biologic treatment for rheumatoid arthritis. Their model classifies the response to biologic treatment with 78% accuracy. It is very high score. However, they used genetic information as an input to the model. That means that it costs a lot to create their machine learning model. Here is our rheumatoid arthritis dataset description, which used in this study. The data includes patients treated at the University of Occupational and Environmental Health in Japan. Items include which biologic products administered, and basic information like age and weight and BMI, blah, 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 and blood test results and formulation information. It's like a drug dosage. And items do not include genetic information. Prediction target is, in this study, is SHIDAI after 6 months. SHIDAI is a composite score which indicates rheumatoid arthritis patient's condition. SHIDAI is calculated from swollen joint count plus tender joint count plus other elements. And The array dataset has many missing parts, so we have to interpret those missing parts. Then we use three different methods: mean imputation, interpolation by sum, and random number imputation. Here I will describe the interpolation method by sum. Sum is a method that models the visual cortex of the cerebral cortex and is classified as unsupervised learning. It allows us to visualize the potential features of the data through nonlinear mapping of high-dimensional data with complex correlations onto a two-dimensional map. Here is a step in missing value estimation and interpolation using sum. First, we train sum using the complete set of samples that does not have any missing parts as input. Next, we calculate similarity between sample vector with missing parts and the reference vector of each node, and assign the missing variables of sample vector with the most similar reference vector. In the last, back to the first step, we iterate these steps until there are no more missing parts. And other two methods to interpret missing parts of dataset are here. Mean imputation. This method imputes each missing value with the mean of observed values. Yes, here imputation by mean of weight. And random number imputation. This method imputes each missing value with random number following a normal distribution. Next, I describe the prediction method. In this study, I use Gaussian process regression to predict rheumatoid arthritis patients' disease activity. GPR is a model to estimate the function from the input vector to the output variable. Estimation of a function is done within the framework of Bayesian estimation, and the output is given as a distribution of functions. The variance is given at the same time as the prediction value, and this variance can be seen as the confidence level of the prediction. In this image, the horizontal axis represents the explanatory variables, and the vertical axis represents the output value of the model. At this time, these blue curves represent the function estimated by the model, and these light blue region represents the estimated function distribution. It can be seen that the variance for the unknown input, right here, is larger than the variance for the observed data. So we think that this confidence level will help physicians to decide whether to rely the prediction models or not. So we choose Gaussian process regression in this study. And next, I will describe the experimental setup. We prepared 3 datasets with different interpretation methods. Dataset 1 is applied mean imputation, and dataset 2 is applied interpolation by sum. And dataset 3 is applied random number imputation. And we divide each 3 datasets into 8 for each biologic product administered, and predict disease activity by Gaussian process regression. And validation is k-fold cross-validation, k-5, and we applied root mean squared error as a metric. And here are the prediction results. Dataset 1 and 2 and 3 are shown in order from the left. And the upper graphs are prediction results for ABT biologic groups. And the lower graphs are ADA biologic groups. The horizontal axis of each graph represents the true value of disease activity, and the vertical axis represents the prediction value. And the size of the points reflect the prediction error. We can see that the higher value of disease activity is, the larger the prediction error becomes. And here are prediction results for the CZP group and the ETN group. Prediction values of the CZP group are smaller than the previous two groups. The ETN group also have smaller prediction values than the true value. And here are prediction results for the GLM group and the IFX group. The IFX group seem to output a prediction value closer to the true value than other groups. However, patients with a large true value, smaller prediction values are output. And here are prediction results for the TCZ and TOFA group. It can be seen that similar values are output for all patients as in the other groups. And for TCZ group, the magnitude of the prediction error is conspicuous. Here is a table which summarizes prediction error by interpretation method and biologic products. The ADA group has the smallest average prediction error, and the TCZ group has the largest prediction error. So regardless of the method of interpretation of the missing parts, the prediction error was almost the same. So as a result, it was difficult to predict disease activity for any of the biologic product groups. Because for CDI, the range of numbers indicating the bare disease activity ranges from 0 to 2.8. All of the prediction errors that I showed previous page exceeded 2.8. So this prediction model is impractical in this study. So here I will try to discuss differences of prediction error between biologic products groups to investigate factors associated with prediction error. I will compare each biologic product groups. To compare each biologic groups, I use the U matrix. The U matrix is a kind of coloring method to color maps obtained after training some. At cluster boundaries in U matrix, the color changes significantly as the vector changes. So we could analyze the map visually. For example, in this image, clusters are here. And first, let's look at the figure of the groups with relatively small prediction errors. These figures show U-matrix representation obtained after training some with our dataset. The black plot represents the location of patients belonging to each biologic product group. And the size of the plot represents the magnitude of the prediction error. And let's look at the figure of the groups with relatively small prediction errors. We can see that patients are distributed all around the map. So there is no bias in the distribution depending on the magnitude of the prediction error. But in the TCZ group, patients had larger prediction errors comparing to the other biologic groups. And TCZ group have more patients densely distributed in the upper parts of the map. And the IFX group, which have the second highest prediction error, also had a higher concentration of patients than the other groups. And it considered that the density of patients in Rheumatoid means that each patient has similar characteristics, and it considered that the estimation of the function in GPR was not successful due to the similarity of patients that lead to an increase in prediction error. However, there is no dense distribution in GRM group, which has the largest prediction error after IFX and TCZ. The size of the prediction error of the GRM group is considered to be due to the small sample size. Okay, here is the conclusion. The purpose of this study is to predict of disease activity in RA patients to support the selection of biologic for treatment. And the experimental results showed that all the biologic groups produced lower values as prediction values in Gaussian process regression than the actual disease activity values. And they had a large prediction error. And there was no changes in prediction accuracy due to differences in interpolation method for missing parts. And from the geometric images, it was confirmed that the biologic groups with a large prediction error formed a dense distribution in their nonlinear space. On the other hand, the biologic group with relatively small prediction error was widely distributed in the geometric map. So in the future, we will explore a more detailed relationship between the prediction performance and the inspection items used as input variables for regression. In addition, we will consider reviewing the prediction model. Thank you for listening.